access of all the subjects anywhere, anytime through the recorded sessions. And also we provide notes with CBMA based question answers, MCQs, and also we provide MCQ from beginner level to expert level, which contain standard multiple choice questions. Then student can also book a slot for personalized counseling session. And here are some subscription plans starting from basic to premium. Basic plan is of 3 months, standard is of 6 months, and premium is of 12 months, which cost around 50,000, and it includes all the services that is provided by Oracle, like pre-recorded class, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module, and also for instant 20% discount, you can use my code Anchal20. So as we have started the marathon exam series, so these are the features. Like in this, we provide rapid revision of all the important questions. Volatile fact are there, and multiple choice questions also will be covered. And also we assure top-notch grades in the exam. And this marathon series is going to be beneficial for all the student, whoever is studying in any university. So we are going to start with the synapse. Okay. So as we can see, what is synapse? Synapse is the junction between two neurons. Okay. Two neurons through which impulse passes from one to another. Okay, so as we can see here, the synapse with another neuron. So this is, here we have neuron 1 and here we have neuron 2. So the transmission is occurring between the one neuron to the another. Then here in neuromuscular junction, we have neuron and muscle. So the transmission is occurring between the neuron and the muscle. Then next we have the neuroglandular synapse. So here the transmission is occurring between the neurons and the gland cells. So synapse is the junction between two neurons, or it can be between neuron to muscle, or it can be between neuron to glands, right? Then based upon the classification, anatomical classification, we have another type of synapse also. For example, we have axonic synapse. Then also we have exodendritic synapse. Then also we have exosomatic synapse. Okay, so what is axonic synapse? So as the name indicates, axonic means in this axon of one neuron is going to transmit on axon of another neuron. Okay, so it's going to be like this. If this is the axon, so the transmission is going to occur between one axon to another axon like this. Okay. Then next we have exodendritic. Okay, so means here axon of one neuron is going to terminate on dendrite of another neuron. So it would be like this. Suppose this is the axon we have. So from here, the transmission is going to occur on the dendrite of the another neuron. Okay, so it will be like this. Then next we have exosomatic. So here axon of one neuron ends on soma. Soma is cell body, okay, of another neuron. So it will be like this. Here we have axon and it is going to end with the cell body. Okay, so it will be like this. Okay, so this is all about the anatomical classification of synapse. Okay, now we're going to talk about the structures. Okay, so very important structures are present. So as already we know the structure of the neuron, it has axon, it has cell body, it has dendrites, right? So another thing that in the synapse we have presynaptic membrane. What is presynaptic membrane? The presynaptic axon terminal has definite intract membrane, and that is known as presynaptic membrane. Then also we have mitochondria. What is the function of mitochondria? It helps in the synthesis of neurotransmitter substances, right? And also we have synaptic vesicles. What are synaptic vesicles? They stores neurotransmitter, very important substance, neurotransmitter. For example, like acetylcholine. Okay. Then also, if we talk about the function of synapse, so in function, it has mainly two functions. The first is basically the main function of the synapse is to transmit the impulse, right? The main function of the synapse is to transmit the impulse, that is action potential, from one neuron to another. But some of the synapse inhibit these impulse, so the impulse are not transmit to postsynaptic neuron, and on the basis of this function the synapse can be divided into excitatory synapse and inhibitory synapse. So as the name suggests, excitatory means the synapse which transmit the impulse, means it does the excitatory function, right? And inhibitory synapse means which inhibit the transmission of impulse, means it does the inhibitory function. Okay, so this is all about the classification, about the structures and about the function. Okay, next we have the mechanism of transmission. So as we can see, here we have the presynaptic neuron, then postsynaptic neuron, then here we have the gap junction, which is also known as synaptic cleft, and here the presynaptic um, the presynaptic membrane has produced the neurotransmitter, for example acetylcholine, and here it has the receptors over which the acetylcholine is going to bind, and the receptors is going to open the channels, okay, like sodium channel, potassium channel, right? So this transmission we're going to study with the help of the flow chart. Okay, so here we're going to study the sequence of events. Okay, so what happened? As the action potential arrives in axon terminal, now what will happen? As the action potential will arrive, then there will be opening of calcium channel, and there will be influx of calcium, right? Now what will happen? This calcium is going to bind with the vesicles, and there will be opening of vesicles and release of acetylcholine into the synaptic cleft. Okay, so these are the very, very important points, and all students have to remember. Okay, so as the acetylcholine is going to pass through the synaptic cleft, now what will happen? There will be formation of acetylcholine receptor complex, right? And also opening of sodium ion and influx of sodium from ECF. Okay. And this influx is going to generate the action potential through the small EPSP. Now, what is this EPSP? This is excitatory postsynaptic potential. Okay, and many EPSP help in the development of action potential, or we can say the summation of EPSP will decide that whether action potential is going to develop or not. Okay, so this is all about the sequence of events that occur in the transmission. Now, next we have the another important topic, that is thalamus. Okay, so when we talk about the thalamus, so thalamus is a very huge part, right? The structure has a very huge number of nucleus, huge parts. So the structure part is only important for the anatomy, but when we talk about the thalamus in physiology, then only the function part is very, very important for the thalamus. So here we're going to discuss about the thalamus only, about the function of thalamus only, and also the question you will get from thalamus in physiology, you will get over the function only. Okay. So what is thalamus? The thalamus is a large ovoid mass of gray matter, right? And where it is situated bilaterally in diencephalon, right? And both thalami form 80% of diencephalon. Okay. Now, talking about the nucleus, as we know, the nucleus is only important for the this anatomy part, but here, as we can see, various nucleus are there. For example, midline nuclei, intralaminar nuclei, like anterior nuclei, dorsomedial nuclei. So various nuclei are there, and also the thalamus is relaying for sensory signal and motor signal to the cerebral cortex, and also responsible for the regulation of consciousness, sleep, and alertness. So all these are the important functions that about we're going to discuss in detail. Okay. So talking about the function of the thalamus. So first, a very important function, that it act as a relay center, right? So thalamus form the relay center for the sensation, right? So whenever any impulse of almost all the sensations reaches the thalamic nuclei, particularly in the ventral posterior nucleus. Okay, so after being processed in the thalamus, the impulse are carried to cerebral cortex through thalamocortical fiber. So impulse carried to cerebral cortex through thalamocortical fibers. Okay. Now, next we have the another function, that the thalamus is the center for processing of sensory information. Okay, so thalamus form the major center for processing the sensory information, and all the peripheral sensory impulse reaching the thalamus are integrated and modified before being sent to the specific area, right? And they are being modified and integrated in the thalamus here. Okay. Now, the function of the thalamus is usually carrying the processing of sensory information. Okay. So this is the main function of the thalamus. Okay. Now, next we have the another function, that this is also the center for determining quality of sensation. So as we know that thalamus is the center for determining the quality of sensation only. Also, that is to determine the affective nature of sensation. Okay. So in a sensation, as we know, they have two quality, the discriminative nature and affective. Okay. Discriminative nature means the ability to recognize the type, the location and other details of the sensation, okay, which is also the function of the cerebral cortex. Whereas the affective nature, it is a capacity to determine whether a sensation is pleasant or unpleasant and agreeable or disagreeable, okay. So this is about the affective nature. Then next, the another function of thalamus is that it is also the center for sexual sensation. Okay, so the thalamus also form the perception for sexual sensations. Then next we have that it has a very significant role in arousal and alertness reactions. So, because of its connection with nuclei of reticular formation also, the thalamus play a very important role in the arousal and as well as in alertness reactions. Then also, this is the center for reflex activity. Okay, so as we know, the sensory fibers relay here, right? This is the relay center. So thalamus form the center for many reflex activities. Okay, so these are the important function of the thalamus, and also the thalamus is also known as the relay center. Okay, as it relay the impulse carried to cerebral cortex. Okay, so Kavya, can you tell me the another name for thalamus?
I
Kavya?
don't know. Can you
Okay,
please explain that?
just now we have studied now that the thalamus is also known as the
English
relay
and we
center.
can continue.
Yes. Why it is known as relay center? Because of the because
Impulse carried in cerebral
yes, because the
cortex.
thalamus form the relay center for the sensation. Okay. And also the impulse of all the sensation reaches the thalamic nuclei, particularly in the ventral posterior lateral nucleus. After being processed in the thalamus, the impulse are carried to cerebral cortex through which fibers? Through thalamocortical fiber. The name of the fiber is very, very important and must to remember. Okay.
Okay, ma'am.
So this is all about the function of the thalamus. Okay, now we're going to learn the another important clinical disorder, that is Parkinson's disease. Okay, so what is Parkinson's disease? This is a slowly progressive degenerative disease of nervous system. Okay, and here this is associated with destruction of brain cells which produce dopamine. Okay, so this disease is associated with brain cells which produce dopamine. Okay, so if the cells which are producing dopamine will get destroyed, so there will be deficiency of dopamine will occur. Okay, so as we can see in the diagram also, the various symptom which is shown by this person. Okay, so as we know, the Parkinson's disease occur due to lack of dopamine. Okay, and caused by the damage of basal ganglia. Okay, so whenever any kind of damage will occur in the basal ganglia, so it can lead to this condition which is known as Parkinson's disease. Okay, and also this is occur due to mostly whenever any kind of destruction is occur in substantia nigra. Okay, so whenever the destruction occur in this part, this can lead to Parkinson's disease. Okay, now we're going to learn that what are the common causes for damage of basal ganglia. Okay, so there are various causes which can lead to damage to basal ganglia, but the most common we're going to discuss. For example, it can be occurred due to viral infection of brain. Okay, like in the case of encephalitis. Okay, in the case of encephalitis, then the next, whenever there is injury to basal ganglia, or third, due to cerebral arteriosclerosis. Kavya, do you know what is arteriosclerosis?
Atherosclerosis is like clot in the heart, blood clot like that type.
Narrowing
Like
of
blood
the
supply, less
artery.
than blood supply.
Yeah, narrowing of the artery. So if it is cerebral arteriosclerosis, means this is occurring in the cerebral part of the brain. Okay. Now, the sign and symptoms, as we can see in the picture also, various sign and symptoms are there, like tremors in the legs, shuffling sound, stiff gait, slightly flexed hip and knees, hand tremors. Okay. And other symptoms are there, like slowness of movement, then rigidity. Okay, stiffness of muscles will occur in limbs, resulting in rigidity of limbs. Okay, then there can be changes in gait. Okay, what is gait? Gait refers to the manner of walking. Okay, and also in Parkinson's disease, the patient loses the normal gait and walks in short steps by bending forward, like this person is doing. Okay, so the person is bending forward and the person will move in the short steps. Okay, then also the Parkinson's disease can be associated with speech problems. Okay, so they speak very softly or sometimes very rapidly. Okay, so these kind of speech problem arise with these patients. Then emotional changes are there. So the person affected by Parkinson's disease may get upset emotionally very rapidly. Okay, so these are the emotional changes. So these are the very common symptoms that you will find in the person who is suffering from this disease. Now coming to the treatment part. Okay, so as we know that this disease occurred due to the lack or due to deficiency of dopamine. So can anyone tell me that what should be the treatment of this disease if the person is suffering from the lack of dopamine? The treatment is simple. Whatever the deficiency, that should get filled, right? So for the treatment, the person should be treated by dopamine injection. Okay, but as we know, the dopamine does not cross the blood-brain barrier. So another substance is also known as L-DOPA. L-DOPA, which can crosses the blood-brain barrier. So it should be injected, and whenever it reaches into the brain after crossing the blood-brain barrier, it will again get converted into dopamine. Okay, so this is all about the Parkinson's. And if the short note over the Parkinson's will come, so all the student need to define the Parkinson's disease. Then what are the means you have to tell, or you have to state the reason of deficiency of dopamine, which substances are destructed. Then also you need to mention the common causes for damage of basal ganglia, then sign and symptoms, and the last you can also mention the treatment. Okay. So this is all about the Parkinson's. Now next we have the another important topic, that is physiology of memory. So what is memory? As we know, memory is defined as the ability to recall past experiences, right? So what is memory? Memory is defined as the ability to recall past experience or information. Okay, and in other words, we can also define memory as the retention or learned material. Okay, now there are various degree of memory, and some memory remain only for a few seconds, some remain for hours, some remains for days, some remains for months, and some remains for years to years, right? Now, when we talk about the anatomic basis of memory, so anatomic basis of memory is the synapse in brain. As we know, the synapse is responsible for the transmission of signals. Okay, so synapse is responsible for memory coding, and here it is slightly different from the other synapse. Okay. So now we're going to learn about the physiological basis of memory. Okay, so whenever we talk about the physiological memory, basis of memory, so as we know, the memory is stored in the brain by the alternation of synaptic transmission between the neurons involved in the memory. Okay. Now the storage of memory may be facilitated or habituated depending upon many factors such as neurotransmitters, synaptic transmission, functional status of brain, etc. So for some factors we're going to discuss. Okay. So for example, first we have facilitation. Okay, what is facilitation? This is the process by which the memory storage is enhanced, and also it involves increase in synaptic transmission, and also it increase the postsynaptic activity, and also this facilitation also known as positive memory. Then next we have habituation. So as the name suggests, this is the process by which the memory stored is attenuated. Attenuated means it get decrease in strength, okay? Basically it get decrease in strength or affect to the value. Okay, it involves in the reduction in synaptic transmission and also slow stoppage of postsynaptic activity. And sometimes it is also known as negative memory. Now, third, we have basis for short-term memory. So the basic mechanism of short-term memory is the development of new neuronal circuits by the information of new synapse and facilitation of synaptic transmission. And also it involves increase in number and size of presynaptic terminals. Now next we have basis for long-term memory. So as the name suggests, basic mechanism for long-term memory is reinforcement of newly formed neuronal circuits resulting in consolidation and encoding of memory in different areas of brain. So this is all about the physiological basis. Now coming to the classification. So as we can see, there are two important form of memory. The first is explicit and second is implicit. So what is explicit? So explicit is basically is defined as the memory that involve in conscious recollection of past experiences. Okay, again I'm repeating, conscious recollection of past experiences. Also it consists of memory regarding events, right, which occurs in the external world around us, and also the information stored may be about a particular event that has happened in a particular time or in a particular space. Okay, for example, the example I can give, the birthday party which the person has attended 3 days before, or the events that taken place while taking breakfast, while taking lunch. So all these things are included in explicit memory. Okay, and also in explicit memory it involves the hippocampus and medial part of temporal lobe. So all the facts and events are included in explicit memory. Okay, now next we have implicit. So here this implicit is defined as the memory in which the past experiences is utilized without conscious awareness. Okay, so the person is not consciously aware, but the past experiences is utilized. Okay, and also it helps you to perform the various skill activity properly, and this all includes procedure, skills and habitats, associative learning, non-associated learnings are also there, and these all are the parts which are involved in carrying or in storing the implicit memory. Okay, so this is all about the physiology of memory and the various forms we have discussed. Another thing we have the condition reflex. So what is condition reflex? Condition reflex is acquired reflex that requires learning, memory, and recall of previous experiences, right? Learning, memory, and recall of previous experiences, and also it is acquired after birth, and it forms the basis of learning, right? So condition reflex is important for forming the basis of learning. Okay, so this condition reflex is always occurred after the birth, whereas the unconditioned reflex is the inborn reflex, which does not need any kind of previous experiences. Okay, now talking about the classification. So in condition reflex, we have a huge classification that we're going to learn. So the condition reflex is classified as classical, conditioned reflex, and instrumental conditioned reflex. In classical, we have the two types: positive condition reflex, negative condition reflex. Okay, so positive means excitation of condition reflex. It also has primary, secondary, tertiary. Whereas in negative, we have external or indirect inhibition, or internal or direct inhibition. Okay, so one by one, we're going to learn about the classification. So starting with the classical part. So as we know, the classical condition reflex are those reflex which are established by conditioned stimulus, okay, followed by unconditioned stimulus. Now we have the various types, like first, for example, we have positive or negative. So positive, in positive, we have the primary, secondary, and tertiary. So primary condition reflex is the reflex which developed with one unconditioned stimulus and one conditioned stimulus, and here the reflex is also established by the condition and with unconditioned stimulus. Both these stimulus should be required, right? Then in secondary condition reflex, it is the reflex developed with one unconditioned stimulus and two conditioned stimulus, whereas in tertiary, a third conditioned stimulus is added and the reflex is established in tertiary positive condition reflex. Okay. Whereas when we talk about the negative condition reflex, so it has external or indirect inhibition and internal or direct inhibition. So in external or direct inhibition, we studied that it is basically inhibited by some form of stimulus, which is quite different from the condition stimulus, and it is also not related with the condition stimulus. For example, some disturbing factors are there, like sudden entrance of a stranger or sudden noise or strong smell, right? So these are included in the external or direct inhibition, whereas in internal we talk about uh, basically there are four ways by which the established condition reflex is abolished by direct or internal factor. The one we can discuss about the condition inhibition. So what is condition inhibition? It's a failure of condition reflex due to introduction of unknown condition stimulus. Okay. When a condition stimulus like flash of light is affected, okay, if another condition stimulus like a ball sound is applied along with the stimulus suddenly, okay, and the response does not occur. So all these included in the internal negative condition reflex. Okay. So this is all about the classical. Then coming to the instrumental condition reflex. So what is this? In instrumental condition reflex, basically they are those reflexes in which the behavior of the person is instrumental. Okay. And in this type of reflex is developed by the condition stimulus followed by a reward or one kind of punishment. Okay. And the instrumental condition reflex are also known as operant condition reflexes. Okay, so this is all about the instrumental and classical condition reflex. Okay, now next we have the another topic of CNS, that is CSF. CSF stands for cerebrospinal fluid. Okay, so what is CSF? It is a clear, colorless and transparent fluid that circulates through the ventricles, brain and subarachnoid space, and also the central canal of the spinal cord. So as we can see, the CSF is present in lateral ventricle, third ventricle, brain, skull, fourth ventricle. It is also present in subarachnoid space. We can see in the picture also, right? So talking about the properties, so the volume of CSF is 150 ml. Rate of formation is 0.3 ml per minute, and reaction is alkaline. The nature is alkaline. Right? And talking about the site of formation, so the CSF always formed in the CSF always formed by choroid plexus. This is also important for MCQ, that CSF also always formed by choroid plexus. Okay, and where this choroid plexus is present, this is present within the ventricles. Okay. Now, talking about the composition, so the CSF contain 99% water and 1% solids. In solids, it has organic and inorganic, both the parts are there. So in organic, it contains protein, amino acid, sugar, cholesterol, urea, uric acid. Whereas in inorganic, it contains sodium, calcium, potassium, magnesium, bicarbonate, phosphate. Various things are there. Now, talking about the circulation. So when we talk about the circulation, as we know, that the formation of CSF occurs within the ventricle. So basically it occurs in lateral ventricle. Now from lateral ventricle through the foramen of Monroe, it enters the third ventricle. And from here through aqueductus salvius, it enters fourth ventricle. Then through foramen of Magendie, it enters cisterna magna and cisterna lateralis. Now from here it enters the central canal and subarachnoid space. And from the subarachnoid space, it enters spinal cord and over cerebral hemisphere. Okay, so this is all about the circulation that occurs of inside the ventricles, various ventricles and spinal cord and over the cerebral hemisphere also. Then talking about the function. So first and the very important function, the protective function. So as we know, the CSF acts as a fluid buffer, okay, and it protects the brain from shock. Okay, so main function that the CSF protect the brain from any kind of shock, and also the specific gravity of brain and CSF is more or less the brain floats in CSF, right? So this is how the CSF perform the protective function. Now next we have the another function that it is also important for the regulation of cranial content. So as we know, the regulation of cranial content volume is essential because brain may be affected, and if the volume of the cranial content increases, it happens in cerebral hemorrhage and brain tumors. An increase in cranial content volume is prevented by greater absorption of CSF to give space for increasing the cranial content. And the last function we have that it has a significant role as medium of exchange. So the CSF form the medium through which many substances, particularly like some nutritive substances and waste material, which also get exchanged between the blood and the brain tissue. Okay, so this is all about the CSF. Now next we have the another and a very small topic, that is blood-brain barrier. Okay, so what is blood-brain barrier? It is a neuroprotective structure that prevent entry of many substances. Okay, so basically blood-brain barrier is present to identify that which substances can pass through it or which substances cannot. Okay, just for example, I will give you a list of those substances which can pass through. Okay, so the substances, just a minute, the substances which can pass through includes like O2, CO2, blue water, then glucose, then amino acids. Okay, so these are the substances. Other are also there. Okay, some electrolytes, some drugs are also there. Then substances which cannot pass through. Just 2, 3 I'm writing here. For example, some injurious chemical agents, various are there, so I'm not going to name anyone. Then pathogens such as bacteria, some drugs. Just for example, I'm writing penicillin. Okay, and also we talk about the dopamine. Dopamine can also pass. Okay, now we're going to talk about the function. The function of BBB, blood-brain barrier. So the BBB act as mechanical barrier and prevent harmful substance entry, right? So this function already we have discussed. Then also it provide healthy environment for brain. How? By preventing the injurious material and organism to enter, right? Then also BBB permit metabolic and essential substance to enter. Okay, and which are those substances? Just now we have discussed like O2, CO2, water, glucose, amino acids, and also other lipid soluble substances are also there. Okay, so this is all about the blood-brain barrier. So I hope you find this class helpful, and also there are more topics are left for these CNS, which are also important, so that we're going to discuss in further class of CNS. Okay, so thank you so much for watching this class.